Hi, my name is Ariel Madrigal. I'm a grad student at McGill University, and my project is named the novo inference of transcription factor motifs from single-cell RNA-seq data. Transcription factors are key molecules. They control cell differentiation and contribute to cellular heterogeneity. Current methods to identify transcription factors in single-cell RNA-seq data rely on previously identified transcription factor motifs. However, nearly 26% of the human transcription factors remain with no known DNA binding motifs. Here, inspired by the prior model, we designed a computational framework that infers de novo transcription factor motifs using single-cell RNA-seq data. The first step of our framework is to generate a Kmer Matrix C. A Kmer is just a DNA sequence of length k. This matrix represents the frequency of Kmers in all the gene promoters of the human genome. Our model exploits the fact that transcriptome data is highly correlated and identify latent variables in the gene expression matrix that represent transcription factor activities. We start with two matrices as input, the gene expression matrix E and the Kmer matrix C. The model decomposes the gene expression matrix into a set matrix that has genes in the rows and latent variables in the columns. Then, set is further modeled as a combination of the C matrix and the activity matrix U. Our method starts using singular value decomposition to reduce the dimensionality of the data. From here, we choose a set of k dimensions that explain a good proportion of the variance. We use the SVT results to initialize set and B. Then we can solve for U using lasso regression. We use block coordinate minimization to solve the system, and we stop when B converges. These are the results of our framework applied to single-cell RNA-seq data from PBMCs. On the left, you can see the results of the EU matrix. This has latent variables in the columns and Kmers in the rows. There are clusters of Kmers showing a similar activity pattern. The idea is that this group of Kmers will represent a given transcription factor or transcription factor family. Then, on the right, you can see the correlation of the actual expression matrix with the one predicted from our model. We can also see how well our model preserves the global structure of the data. On the left, you can see a UMAP performed on the actual expression matrix. And then, on the right, you can see a UMAP performed on the B matrix that relates latent variables to cells. As you can see, the output results maintain the structure of the data, keeping the same clusters. For the motif reconstruction, we will start from the results of matrix U. Remember that matrix U has Kmers in the rows and latent variables in the columns. We'll cluster the Kmers based on their activity profile, and then also on their sequence similarity. Once we define groups of related sequences, we'll be able to perform de novo assembly to reconstruct DNA binding motifs. Although we are still working on tuning some of these steps, we have some encouraging results. On the left, you can see a UMAP of the distance matrix of Kmers based only on sequence similarity. The cluster labels come from the clustering performed on the activity matrix U. Interestingly, cluster 23 comes together based on activity and sequence. And then, on the right, we show that some of our latent variables in the columns correlate with the expression of transcription factors in the rows in the dataset. Regarding our conclusions, we show that using an integrated model that considers gene expression, a set of latent variables, and the Kmer matrix, we can detect Kmers that correlate with the specific transcription factor signatures. We also show that our reduced latent variable dimensional space preserves the structure of the data. Some of the future steps that we want to perform are to implement the de novo assembly steps using the cluster Kmers. And then we want to apply their method to data derived from the human cell atlas. Finally, I want to say thanks to my supervisor and to all the members of my lab, also to the Human Genetics Department at McGill, and especially thank you for listening.